and welcome. It's an honor to be able to introduce our first speaker, Dr. Matt Lucy, who is a full professor at the University of Missouri, which is just about 120 miles west of us. I think I was picked because I'm tall, loud, and obnoxious, and you'll all have to listen to me for just a few minutes. Dr. Lucy is a graduate of Cornell University. He then went on to Kansas State and worked with Jeff Stevenson for his master's, and then he earned a PhD down in Florida under Bill Thatcher. So he has quite a illustrious pedigree, if some of you older people would know those researchers. His current research is it involves endocrinology and dairy cattle. And although he's not speaking about dairy cattle fertility today, he's really quite internationally famous for his work in fertility issues in dairy cattle. I'd like to just have you read the full biography because I'm not going to go over it, but I think among the awards that Dr. Lucy has earned, I think perhaps the most important one is that the American Society of Animal Science named him or gave him the Animal Physiology and Endocrinology Award in 2010, and he's currently the editor-in-chief of the Journal of Dairy Science. Now, I had the privilege of working with Matt some years ago on the effects of somatotrophin, or if you please, growth hormone on both superovulation and recipient pregnancies in cattle, and I can't remember quite when that was, but it was published in Theriogenology. And on a personal level, he's not all science. Matt and I have fished for bass here in Missouri. We have fished for trout in New Zealand, and we're going to go fish for salmon in Alaska one of these days. But without further ado, Dr. Lucy, it's all yours.
All right, thank you very much. Thank you. Thanks, John. That was fantastic. Yeah, John is a good friend, and I appreciate the introduction, and I also appreciate the invitation. And I want to talk to you a little bit today about, we're going to talk about dairy cows, and of course, dairy guys like me, we always start out with high production cows, and you may or may not know, but there's a new record holder in terms of production for Holstein, and that's this cow, Gigi, out of Wisconsin, that produced about 75,000 pounds of milk in a single lactation. And that's a big number, and then I started thinking, well, okay, so if a cow is producing that much milk in a single lactation, exactly how much milk is that a day? Well, Gigi, when she was at peak production, was probably producing 30 gallons of milk each day, which is quite amazing that an animal could produce that much milk in a single day, but that's what that record basically translates into. And the other thing that's really interesting, and where I'm going to take this talk, is the fact that it takes 72 grams of glucose to make a single kilogram of milk. And 72, and for somebody like Gigi, that equates to about 8.3 kilograms of glucose each day, or about 16 pounds of glucose. And I realize that these bags contain sucrose, and not glucose, but you get the idea. And the reason that's so interesting is because, we're dealing with a ruminant, and very little glucose actually leaves the digestive tract of the ruminant. So it's fermented and metabolized. So that means that this cow must synthesize, in this case, 8 kilograms of glucose per day in her liver. And this is a tremendous metabolic issue for a cow postpartum that's producing quite a lot of milk. And so this graph shows glucose concentrations in the cow, and what it's showing here is this is from minus 10 to 20 days after calving. And so, the peak here is the day that the cow calves, and there's quite a bit of glycogen stored in the cow's muscle and liver, and on the day of calving, she releases that glycogen. And what we see is after she calves, the circulating glucose concentrations actually decrease, and this reflects the fact that she is basically strained to produce adequate glucose to meet the needs during lactation. So these are data from a study by Dr. Garvik and I did, and what we did was sorted the cows based on whether they got pregnant to first insemination or didn't get pregnant to first insemination. And what is interesting about this is glucose, as well as some other metabolites, but glucose clearly is different during this period in cows that will eventually become pregnant or not become pregnant. And actually, this relationship is quite strong. So there's something that happens early postpartum in a cow related to her metabolism that determines whether or not she'll get pregnant 60 to 100 days later. And we're not the only ones that have this kind of data. These are data from Ireland for high fertility and low fertility cows, and these are glucose concentrations. And again, what you can see, the blue is the high fertility animals have elevated glucose compared to the low fertility animals. The other thing you can see in this graph, which is true from our data, is that really these metabolic differences that determine fertility, they're all clearly at play during the first 3 weeks postpartum. That's when things are really happening that affect the long-term fertility of the cow. It's not so much later when we're breeding the cow. It's really these things that are happening right around calving in the first few weeks after calving. So that got us thinking about this process of uterine involution. So what is going on, or what's happening in the cow during the first 3 weeks after calving that might ultimately affect whether or not she'll get pregnant later on? And of course, well, she's returning to cyclicity, and we're familiar with that story. But the other part of this story is the fact that her uterus is undergoing involution. And this is a picture of a cow's uterus 3 days after calving. This cow has successfully passed her placenta, but nonetheless, if we look inside the uterine horn, we can see that the caruncles are still present. These have to be sloughed off and broken down. And as well as you can understand, this uterus has to shrink back to a normal size. This is a meter-long stick. And the process that the cow uses during uterine involution is an inflammatory reaction. So inflammation is the process that's responsible for uterine involution in the cow. So it's absolutely necessary, and inflammation is a good thing. But I think we all realize as well that if inflammation goes on for too long, it becomes a bad thing. And that's certainly true for what happens inside the uterus of the cow, because we know that diseases like endometritis, which are a prolonged period of uterine inflammation, clearly affect whether or not the cow gets pregnant. And so that got me thinking a lot about, okay, inflammation, and what are the implications of inflammation in terms of uterine health of the cow postpartum. And to show you what inflammation looks like, I just want to show you a cross-section of the uterus from a heifer. And when we look at a cross-section of the uterus from a heifer, you can see the endometrium, and you can see the lumen of the uterus. And of course, the lumen of the uterus is where the conceptus develops. And you can see the glands that secrete things that feed the conceptus for its growth. And if we take a higher magnification, you can see the glands again, and you can see blood vessels and the luminal epithelium, which lines the uterus, and you can see fibroblasts, which form the stroma, and you can see a single immune cell shown there. So it's not unusual to have some immune cells within the stroma of the uterus, and this is perfectly normal. Now, if we take a look at what the uterine lining looks like of a cow at 25 days postpartum after calving, you can see the luminal epithelium and you can see the stroma, but you can see that the stroma is packed with immune cells, and this is inflammation. And these immune cells are there to deal with not only the pathogens inside the uterus, but they also are responsible for breaking down that tissue and getting that tissue out of there and restoring the uterus to a normal size. And one of the key immune cells, of course, are the neutrophils, and in this case, you can see the neutrophils pushing their way through the luminal epithelium in an attempt to get to the lumen and deal with not only pathogens, but also this other material that needs to be digested and basically broken down. So if you look at what's going on inside the uterus of a cow at 25 days postpartum, it's actually, as I said, highly inflamed. But you also get the sense when you look at a lot of histological sections that this whole process of inflammation and these neutrophils, it also appears to really be damaging the uterus. And in this case, you can see that a piece of the luminal epithelium shown here is actually being torn away by these neutrophils trying to get to the center of the uterus. And so you immediately get the sense that if this process this inflammatory process goes on too long, it might be quite damaging to the uterus and could potentially ultimately affect the long-term fertility of the cow. So this is kind of how we got into this whole area and got interested in this. So we in our lab, we do take every opportunity we can get to look at the uterus of the cow within 1 month after calving. And it's kind of a tricky time to get material because in general, people don't like to kill dairy cows 1 month after calving. I mean, they're entering peak lactation. It's not the greatest time. But nonetheless, we take every opportunity we can. And typically what we do in my lab is I just send the students out and they get the uterus and they take pictures of the inside and they take samples for histology. And one of the things that was really interesting, I was reading their notes, and they kept talking about these warts on the lining of the uterus, and these warts. And I said, "What do you?" So finally, I said, "What are you talking about, warts?" And they showed me these pictures of these warts, and I said, "Well, what is that thing?" And actually, what's really interesting is those are pseudopolyps that form on the lining of the uterus of the cow postpartum. And what pseudopolyps are they're typically described in ulcerative colonitis in humans, and these are products of inflammatory reactions. And in colonitis, they slough off the colon, and this leads to bleeding and ulcerative colonitis. And in cows, basically, this is a histological section of one of these pseudopolyps in the cow, and this is a histological section of one of these pseudopolyps in the human. And if we look at these under higher magnification in the cow, for example, you again can see the luminal epithelium on this pseudopolyp, and you see this tremendous, it's just absolutely packed with immune cells. And this got me to thinking about, when we think about ulcerative colonitis in people, we think about this as an inflammatory disease, and we approach it in a method to try to cure inflammatory diseases. But we don't necessarily, although the uterus of the cow appears to be having some similar pathology, we don't necessarily think about it as an inflammatory disease or approach it in a method to try to treat it as an inflammatory disease when it occurs. And this really has got my interest, is how do we really deal with metritis and endometritis as an inflammatory disease instead of as however we look at it today. So the thing that's really interesting about inflammation in the uterus is that, as I mentioned earlier, inflammation is a good thing and inflammation is a bad thing. The good part of inflammation is if we look at other tissues that are undergoing tissue regeneration after injury, inflammation is absolutely essential because those immune cells that come into that tissue release cytokines or release hormones that are important for stem cells and stem cells within the uterus to develop and repair the uterus. So these immune cells are important to the reparative process. So it's probably absolutely essential that this inflammatory reaction goes on so that these cells that are rebuilding the uterus can be stimulated. And so if we look at what happens in the uterus, this is a very high power magnification of the deep glands inside the uterus. And what happens in the uterus postpartum is that, this is on day 30 postpartum, is that the cells deep in the uterine glands begin to divide. And you can see here the arrows show mitotic figures deep in the glands. And these cells are dividing, and as they proliferate, they move out of the glands and restore the luminal epithelium on the outside of the uterus. So this is, again, a section of the uterus at 30 days postpartum. You can see it's packed with inflammatory cells. You can see that the luminal epithelium here, and here's one of these luminal epithelial cells, the arrow. Here's one of these luminal epithelial cells that is stretching to attempt to cover or repair the luminal epithelium in the postpartum cow. And so what I got to thinking about was, okay, sometimes this process goes really well, this involution process. And this is a picture. It's taken at the exact same magnification, let's say, as the picture on day 3. And this is a uterus 25 days after calving. And you can see that, wow, that looks pretty darn good for that's 22 days, or about 3 weeks after that first picture I showed you. And that looks pretty darn good. But we also know that sometimes this process goes very badly. And this is another uterus at 25 days postpartum. And you can see that there's quite a lot of blood here. And I think that's probably from the ulceration from those pseudopolyps. There's quite a lot of blood and also quite a lot of pus in this uterus. And I guess what I got to thinking about was, I wonder what the implications are in terms of those cells that are regenerating during this process. I mean, what's the implications of the fact that this process is going so well in one cow and so poorly in another cow? And if we look at this, and if we look at the scientific literature, what we understand is that cells have what's known as an epigenetic memory. In other words, they remember what happened to them. And the question I began to think about is, do these cells that are regenerating the uterine lining and repopulating the uterus, do they remember where they came from? Do they remember the fact that this entire process went very badly, for example? And that's kind of where we were thinking about this. The traditional methods, I think, as people understand, the traditional methods for treating cows that have metritis, for example, typically involve some sort of antibiotic treatment. And the antibiotics that are used globally are different. The approaches are different. So, for example, in many parts of the world, it's intrauterine cefepirins, and this is data from that, in which they've treated these cows with, this is the Metricure product. And you can see this intrauterine cefepirine does not improve the fertility of cows that are healthy, but in general, cows that have either a vaginal discharge or endometritis based on cytology, generally this treatment, at least in this study, improves fertility at first insemination. But I don't know about you and your practices, but many of the vets that I talk to, this is still not necessarily wholly accepted that simple antibiotic infusion into the uterus will ultimately restore fertility in cows with metritis. I guess that's what I'm trying to say. There are some studies that suggest that this helps, but certainly in the United States, we don't have any approved antibiotics for intrauterine infusion. What we do have is ceftiofur, but ceftiofur is only labeled for the treatment of the disease. It's not labeled to restore fertility. So if you look at a lot of data on the fertility of cows that have metritis postpartum, what you find is that if we compare healthy control animals on these survival plots, if we compare healthy control animals to cows that have had metritis, what we see is, regardless of how you treat them, they never achieve the same fertility postpartum as a cow that didn't have metritis. So for whatever reason, these cows that have metritis within 1 week after calving never come back to the same fertility, in some cases after 200 or 300 days. And that's the question. I mean, why don't they get pregnant? I mean, why doesn't fertility come back to normal? I mean, what exactly is going on inside these cows that leads to long-term infertility? And so inflammation is important. We expect a rapid inflammatory reaction inside the uterus. And I think we also believe that prolonged or protracted inflammation inside the uterus leads to infertility. So that brings me back to glucose. And as I mentioned, we think that glucose is a very important substrate in cattle, and it's important for the reproductive biology of the cow. It also controls things like insulin and IGF-1, which are important for reproduction. But it's also, again, a difficult metabolite for cows to deal with because they basically have to synthesize all this glucose in the liver, which then circulates in the cow. And the reason that glucose is so critical for the immunology of the cow is that these immune cells that are in the business of cleaning up the uterus and removing pathogens from the uterus and removing debris inside the uterus, these guys use glucose as their primary energy source. So the question in my mind was, well, maybe the reason this whole process doesn't work real well on a cow is because there's not enough glucose and they don't have enough energy to do their work. And in fact, if we look at these cells, these immune cells, and we look at the things that make them happy, that make them function well, they really like elevated high glucose, high insulin, high IGF-1, low BHBs, or low ketones, and low NEFAs. Well, if you think about what we're dealing with in a cow during the first week postpartum, we're dealing with low glucose, low insulin, low IGF-1, high ketone concentrations, and high NEFAs. So basically, metabolically, the things that we know that support milk production in a cow, the metabolites and hormones, are exactly opposite to what these immune cells that are responsible for uterine involution or cleaning up the uterus during uterine involution, they're opposite to what those immune cells want. So one possibility is that basically they don't function well because of this metabolic profile. So in 2013 to 2014, I was in Ireland and I did a sabbatical there, and what we were working on was this glucose question. Okay, well, if it's all about the glucose, let's just infuse glucose 24 hours a day into a cow, postpartum cow, and ask the question, do these neutrophils function better? Okay, so that was a hypothesis. If we just give them glucose, can we fix this problem? And as Thomas Huxley said, you know, this was one of these great tragedies of science, the slaying of a beautiful hypothesis by an ugly fact. And boy, did this hypothesis get slayed, because this is the data for neutrophil function. We're looking at oxidative burst during 7, 14, 21, and 28 days postpartum. Cows infused 24 hours a day with glucose or control. And you can see the improvement in the function of the neutrophils over time, and we know they do improve function over time, but you can also see that normalizing glucose had no benefit whatsoever to these guys. So There are, I don't know if you guys are following the ImRestore story. I don't work for Elanco, but I'll just mention ImRestore. Elanco just started marketing ImRestore, and ImRestore is a recombinant granulocyte colony stimulating factor. And basically, I think the protocol is you treat cows once prepartum, and then treat them 3 days postpartum or something along those lines. And what ImRestore is specifically designed to stimulate neutrophil function. This product is designed to basically do what we were attempting to do with the glucose infusion. And so it's labeled for mastitis prevention, but mastitis is associated with poor immunology postpartum, and these neutrophils are also what's responsible for dealing with mastitis. So the question is, we'll have to wait and see whether this also has some application in dealing with metritis. But this product is designed to address this issue, and that's where it's at. So the other part of this work that we were working on that related to glucose was the fact that, okay, well, maybe not so important early postpartum, but maybe important later postpartum when we're trying to breed the cow. Because actually, the early embryo and the developing conceptus really depends on glucose as an energy source for growth. So, okay, well, maybe the reason we deal with infertility in cows has something to do with these low glucose concentrations as well. I mean, this is possible. And so we had a hypothesis that stated that, well, we've got glucose, and a lot of this glucose gets diverted to the mammary gland, and that leaves less sort of in the maternal glucose pool and less for the developing conceptus. So we said, well, maybe this is creating problems, this low glucose for the conceptus. And so we did a tremendous study, and this was with one of my students, Tyler Stratman, who just graduated. And we measured embryos, and we looked at glucose concentrations. And so these are data for growth of the embryo between 32 and 45 days in cows with high glucose or low glucose. And the bottom line was, yeah, maybe a tiny effect on the growth of the embryo, but actually not big enough to concern yourselves with. I mean, the embryos seem to survive normally regardless of the circulating glucose concentrations in the mother. And it didn't, when we saw embryonic loss in these cows, when embryos died, that was clearly a catastrophic event. And low blood glucose was not causing these catastrophic events. So we couldn't really associate anything metabolically with the cow with how fast the embryo was developing during this period. So that hypothesis went away. So there was this period in my life where everything you test goes to dust. And so you end up with just like, why? Why am I in science, right? Why am I in veterinary medicine, okay? Why am I in science? So I was left with this, why are these differences in glucose right around calving important? And, why do cows that have metritis end up never getting pregnant, or they're so difficult to get pregnant even 200 or 300 days later? And why doesn't that work? And why do we have embryonic loss? And about this time, there was a lot of data coming out on the microbiome, and I found it really interesting, this concept that the microbes that live on us and in us can potentially have implications in terms of health. And the data, they say there's the bacteria, there's 10 times more bacteria living on you and in you than actually you have in terms of cells on you or in you, your own cells. And so there's large populations. And so they're found in lots of different places. In the reproductive tract, they're traditionally described in the vagina and in the cervix. And there's just been a real explosion of research in terms of this microbiome. And I started thinking about the microbiome in the cow and wondering, okay, is there something I need to understand about the microbiome that affects the cow's fertility? And then there was this paper published in Science talking about there was a microbiome in the placenta. And I'm thinking to myself, like a microbiome, like bacteria living in the placenta during a pregnancy? That sounds interesting. So I did what every good animal science guy does. He goes over to the vet school and he talks to his buddies there, and Dr. Volkman at the University of Missouri, and he was doing a C-section, and I said, can I get some placenta and some meconium and I'll get some feces from the adult and some rumen from the adult, and let's send it over for metagenomic sequencing, which is basically you send it over for DNA sequencing, but they're sequencing the bacteria that are there. And lo and behold, as you would expect, these bars are different bacterial species, so they represent different bacterial species, up to 100%. And as you'd expect, there's tons of bacteria in feces, and there's tons of bacteria in rumen. But there's also there is a bacterial community that live in the placenta and also the meconium. Now, the meconium probably shouldn't have been that big a surprise. I mean, people have known that in meconium there is a population of bacteria in the gut at birth, at calving in this case. But the placenta was interesting. And it got me to thinking about, exactly what sort of bugs are in the placenta. And is it possible that some of these bugs that are in the placenta are same bugs that are causing metritis postpartum? And let me first say that there's no question that environmentals are a major part of the environmental pathogens at calving are a major part of the picture for metritis. There's just absolutely no question about that. But I started to wonder, well, maybe in addition to environmental pathogens, maybe there's pathogens that are in the placenta already, that are already there, and then when the cow calves, for some reason, some cows have no problem dealing with this, and other cows don't deal with it well and get metritis. I mean, that was the idea. And what was interesting, these are data from Martin Sheldon's, a review from Martin Sheldon. And when we looked at what was what was there, in terms of the real bad guys, what we saw in the placentome was Prevotella. So we didn't see the other bad guys, but we did see Prevotella in the placentome, which is one of the bad guys. And then we saw a lot of the other potential pathogens. So it appeared that, okay, not all the bad guys are there, but Prevotella seems to be there. And so since that time, we went back, and we're, again, working with my friends in the College of Veterinary Medicine at Mizzou, Scott Polk and Pedro Melendez, and we work with Aaron Erickson at the MU Metagenomics Center. And so we went back and examined additional pregnancies. And in this case, we went to a slaughterhouse. That was one pregnancy. Okay, that's not so great, right? That's N equals 1. But nonetheless, we went to a slaughterhouse, got 5 pregnancies in third trimester, and collected tissues from 5 pregnancies in the third trimester. And the tissues we collected a control tissue from the outside of the tract. So this was at a slaughterhouse, so we got a control sample from the outside of the tract. And then we collected samples from cervix, intercaruncular placenta, fetal fluids, as well as, intercotyledonary placenta, fetal fluids, as well as placentomes. And again, we subjected them to sequencing, and again, we found that that there were bacteria in there, which is interesting. So if we look at, this is a pCNA plot, and what this basically does is it's a way to look at the entire population of bacteria and group it together. And what we saw is that the control samples grouped together. And so that's good. That was the stuff that is basic. Those are basically environmentals that found themselves on the outside of the tract as it was moving through the slaughterhouse before we were able to collect our samples. The cervical samples also clustered together, and that makes sense too because we know there's a microbiome inside the cervix of the cow. But then the samples from the placenta and the fetal fluids also clustered together, indicating that the microbiome in those samples is different. Now, I want to emphasize, I'm not suggesting there is a raging infection inside the cow's uterus. I'm not saying that this is necessarily a disease state, but I am saying that these organisms potentially are living there, and we don't really understand what they're doing, and we don't really understand what the implications for them are as a cow goes from the pregnant state to the non-pregnant state and attempts to progress through uterine involution. So we actually took this a little bit further, and in this experiment, we looked at, well, said, okay, well, if there's something there in pregnancy, is there something there in the virgin heifer? And when does this thing get established? I mean, when does this microbiome enter the uterus? One possibility is it might enter the uterus when we first breed a heifer. I mean, we take an AI rod, we run it through the vagina, we run it through the cervix, and that may introduce the population at that point. So we just don't really know at this point. But in this experiment, we took 10 virgin heifers, and we also took 32 cows at 1, 5, and 9 weeks postpartum. And we sampled them, all these animals were sampled through a transcervical approach with guarded sampling devices. We did the best we could to ensure that we weren't simply dragging bacteria through the cervix into the uterus. So with that caveat. And again, these are PCNA plots, PCA plots, which attempt to show the relationships of these microbiomes between individuals. So the first point is that if we look at the heifers, they cluster very tightly together, and they are significantly different than any of the cows. So whatever is inside the heifer uterus, those aren't necessarily the same bugs or pathogens. I don't want to use the word pathogens because these are not necessarily pathogens, but they are not necessarily, it's not the same population as you see in a cow. This big circle shown here, that's the cows, this big circle shown here, those are the cows at 1 week postpartum. And we do know that there are lots of bacteria in the uterus at 1 week postpartum. But then if we look at 5 and 9 weeks postpartum, you can see that at 5 and 9 weeks postpartum, we're really dealing with a different microbiome at that point. In other words, things have evolved. So the question that I have is, things have evolved, and does it matter how they have evolved? Does this affect what is there at 9 weeks postpartum about the time I'm getting ready to inseminate this cow? Does that have implications in terms of whether or not she'll get pregnant? And if the wrong guys are there, does that have long-term implications in terms of whether or not the cow gets pregnant? So this, following on, and I'll finish briefly, is that, so in terms of the microbiome, that's what we've been thinking about. What are the implications of this microbiome? Does the presence of certain bacteria lead to sort of prolonged inflammation in the cow? And does that prolonged inflammation in the cow affect her long-term capacity to get pregnant? So at about the same time we were doing all this microbiome research, we published this paper in Theriogenology. We just published it, actually. And it had to do with these things, again, inside the uterus of a postpartum cow. And it's a long story, but quite a while ago, Zoetis sponsored some research, and we were studying the development of the conceptus, and we were slaughtering cows at 28, 35, and 42 days of pregnancy. And this was sponsored by Zoetis. And we did histology on these cows, postpartum dairy cows, 28, 35, and 45. And I noticed all these clusters of immune cells inside the uterus, and these are lymphocytic foci of these cows that we slaughtered that were pregnant. And I thought, you know, what's the deal with that? I mean, what are those foci doing in these cows that are pregnant? I find that, I mean, why are they there? And these foci, these foci, they come in different sizes. So there's some of them are sort of medium, some are very large, and some are small. And if you do the immunohistochemistry on them, what you find is they're actually, these foci are T cells surrounding a core of B cells. And if you look at a cross-section of the uterus, now this is at a very low magnification, and you can see a caruncular area, an intercaruncular area. You can see that the brown spots are all these foci. Well, that's interesting. That's a whole lot of immune foci in the cross-section of a cow's uterus. And, of course, I'll continue on with the story. So you see all these foci. And the other thing that struck me is that we had, in this experiment, we had 43 cows on the experiment. There were 11 that had evidence of some sort of uterine infection postpartum. And clearly, the ones that had a uterine infection postpartum had more of these foci later on. So there was an association between the presence of these foci inside the uterus and this early uterine disease. So I was thinking about these foci and what they're doing and reading about the gorilla genome and science. And don't ask me why I was reading about I don't know what that says about you, John. But no, just kidding, John. But poor John, you didn't think you were going to get through this whole thing without me harassing you somewhat. But anyways, and there in this science magazine, I turned the page and there's this huge story, immunity goes local. And I find out that lots of people are studying these things. And they're studying them because they don't really understand what are the implications of these things. So these foci, they tend to set up shop. They're T cells and B cells. So the idea is the T cells are recognizing antigens and communicating the information to B cells, and the B cells are producing antibodies. And it's a local sort of unit that's dealing with whatever, whatever pathogens or whatever is going on there. And of course, but lots of people are really interested in these. I mean, are they a good thing? Is it good? Is it maybe a local little unit that's really critical so that if something shows up, they're ready to fight it? Or is it bad? Does that mean that there's something already there? And people don't really understand. Now, but they use different words. In the human literature and the mouse literature, they call them tertiary lymphoid structures, whereas we would call them lymphocytic foci in the veterinary community. The thing that was interesting about this was that in this experiment, we were slaughtering these cows on 28 and 35 and 42 days of pregnancy. And you can see that if we group the cows based on whether or not the dark bars was cows that had a lot of these foci, the clear bars are cows that don't have so many. We group these cows. You can see that things progress pretty well, but then we get out to about 42 days of pregnancy, and the placenta slows down in terms of its development. They are associated with inflammation. And it's the same sort of story. There's an associated inflammation, so cows that have a lot of these foci also have inflammation, and you can see the effects of the inflammation on the development of the pregnancy. And we're running a little bit short on time, but I'll just say that we had some cows that had embryonic loss, and when we looked at those cows with embryonic loss, we opened up the tract and found a dead embryo. They generally had a lot of these foci and a lot of inflammation. So when we think about this, of course, for you practitioners that are familiar with the equine literature, of course, the horse guides have known this for a long time. Inflammation leads to embryonic loss, particularly later between day 60 and 90 of pregnancy. But in the bovine literature, it's not so well established that inflammation leads to embryonic loss. But and of course, these foci were well described in horses, as well as the associated inflammation. But it appears that perhaps the same thing is going on here, where for whatever reason, when this embryo that's developing encounters inflammation or encounters foci, for whatever reason, it dies. And that's where we are in terms of the science right now. So I just will conclude by saying I think this microbiome is really going to be interesting. I think it's interesting in terms of implications for innate immunity. I think it's interesting and it has implications in terms of adaptive immunity. But I think the other thing that's really going to be really interesting is questions about, okay, if there is a microbiome inside the uterus, are there good microbiomes that are perfectly happy, are happy? Or are there bad microbiomes that are unhappy? And one of the things that you wonder about is when we treat with antibiotics, for example, when we treat uterine infection with antibiotics, what are the implications in terms of that good microbiome and that bad microbiome? And how does that affect the interactions between those two groups of bacteria with some of our treatments? So hopefully where this will all lead to is maybe a better understanding of exactly what's going on and maybe better ability to treat metritis so cows don't end up infertile for 200 to 300 days. So with that, I'd like to say thank you very much. Happy to ask any questions. Thank you.